Today we're going to be talking about cell membranes. Since the invention of the microscope in the 17th century, it has been known that both plant and animal tissue are composed of cells. The plant cell wall was easily visible even with this early microscope, but no similar barrier was visible on animal cells. But surely some sort of barrier must have existed. By the 19th century, this question was being actively investigated. Moritz Traube noted that this outer layer must be semi-permeable to allow transport of ions. Not that kind of iron. Traube, however, had no evidence for this. The lipid nature of the cell membrane was first correctly intuited by Quink, who noted that a cell generally forms a spherical shape in water. The only other known material to exhibit this behaviour was oil. Based on these observations, Quink asserted that the cell membrane comprised a fluid layer of fat less than 100 nanometers thick. By the early 20th century, the chemical but not the structural nature of the cell membrane was known. Two experiments in 1924 laid the groundwork to fill in these gaps. By measuring the capacitance of the erythrocyte solutions, Frick determined that the cell membrane was 3.3 nanometers thick. Frick, however, misinterpreted the data, which led him to believe the cell membrane was a single molecular layer. This is because the polar lipid head groups are fully hydrated, so they don't show up in a capacitance measurement. So in actual fact, this experiment only measured the thickness of the hydrocarbon core, not the whole bilayer. Soon after, two people named Gorter and Grendel approached the problem from a different perspective. They performed a solvent extraction of the erythrocyte lipids and spreading the resulting material as a monolayer. When comparing the area of the monolayer to the surface area of the cells, they found a 2-to-1 ratio. There was a few problems with this experiment, but them aside, the conclusion was that the cell membrane is a lipid bilayer. A decade later, and Davison and Danieli proposed a modification to this concept. In their model, the lipid bilayer was coated on either side with a layer of globular proteins. But like the many before them, their theory was also incorrect. After a little while, a more direct investigation of the membrane was made possible through the use of electron microscopy. After staining with heavy metal labels, two thin dark lines separated by a light region were noticed, which was incorrectly interpreted as a single molecular layer of protein. A more accurate interpretation was made by David Robertson, where he suggested that the protein layer is formed from a monolayer of polypeptide chains rather than the whole protein molecule. We now move on to how we know the cell membrane today. Today the membrane is represented in a model called the fluid mosaic, which was created by Singer and Nicholson. There was again a development of Daniela and Davidson's model, but this time, compared to Robertson's model, it had significant differences. The key differences were the proteins don't form a structural layer holding the lipids in place. The proteins are not attached to the outside of the lipid bilayer, but embedded within it. And this is what we know the cell membrane as today. Thanks for watching.